This is a piece by Steve Mirsky, who's a columnist for Scientific American, and he wrote this after the whole Cobb County, Georgia thing about putting stickers on science books regarding evolution. And of course that's all been resolved, but this is still relevant, and it's funny, and it fits in with today's topic, so I thought it'd be pretty good. So this appeared in the February 2005 issue of Scientific American. And it's called Sticker Shock. In the beginning was the cautionary advisory. Brush fires are raging all across America over the teaching of evolution, as various anti-evolution interests attempt to give religiously based views equal footing in science classes. These fires are fueled by so-called creation scientists who allege that they have scientific evidence against evolution. They don't. Their co-conspirators, the intelligent design crowd, go with the full-blown intellectual surrender strategy. They say that life on Earth is so complex that the only way to explain it is through the intercession of an intelligent super being. They don't mention you-know-who by name as the designer, but you-know-who is. And it isn't Brahma. One little blaze can be found in Cobb County, Georgia. As this issue of Scientific American went to press, a federal judge in Atlanta was in the process of deciding whether biology textbooks in the county could continue to sport a warning sticker that read, "This textbook contains material on evolution. Evolution is a theory, not a fact, regarding the origin of living things. This material should be approached with an open mind, studied carefully, and critically considered." Maybe that last sentence should be stamped into every textbook, and some other books I can think of. And maybe they could rewrite the advisory so that it's accurate, perhaps something like, "Variation coupled with natural selection is the most widely accepted theory that explains evolution. Evidence for evolution itself is so overwhelming that those who deny its reality can do so only through non-scientific arguments. They have every right to hold such views, they just can't teach them as science in this science class." But why pick on evolution in the first place when there's so much to be offended by in virtually any science class? I propose that Cobb County-style stickers be placed in numerous other textbooks. Here are some suggestions. Sticker in Introduction to Cosmology. Astronomers estimate the age of the universe to be approximately 13 billion years. If evolution ticks you off because you believe the Earth is only 6,000 years old, cosmology should really make smoke come out of your ears. There's a fire extinguisher next to the telescope. Sticker in Geography for Today. Some people believe that the Earth is flat. An ant probably thinks the beach ball he's walking on is flat too. Anyway, this book says the Earth is more like an oblate spheroid. Now go find Moldova on a map. Sticker in Earth Science. You are free to exercise your First Amendment rights in this class and to identify all stratigraphic layers as being 6,000 years old. We are free to flunk you. Sticker in Collegiate Chemistry. Electrons. They're like little tiny ball bearings that fly around the atomic nucleus like planets orbit the sun. Except that they're actually waves. Only what they really are probability waves. But they do make your MP3 player run, seriously. Sticker in our solar system. Remember they said in chemistry class that the electrons fly around the nucleus like planets orbit the sun? Some people think the sun and other planets go around the Earth. You'll have a much easier time with the math if you just let everybody go around the sun. Trust me. Sticker in Creationism for Dummies. Religious belief rests on a foundation of faith. Seeking empirical evidence for support of one's faith-based beliefs, therefore, could be considered pointless or even blasphemous. Sticker in Modern Optics. Caution, dark ages and mirror may be closer than they appear.
HGP is fortunate in the wealth and variety of expertise in its own membership. And Dr. Duane Ray's professional background in physics makes him our go-to person for science questions. He has given presentations for us on the Big Bang Theory, on its antithesis, creation science, and on quantum mechanics and relativity. Today's talk should bring us up to speed on DNA, evolution, and other undisclosed biological stuff. We have probably all heard the story of Rene Descartes ordering a hamburger, yes? When the waitress inquires, "Would you like fries with that?" Descartes says, "I think not," and instantly disappears. witticism illustrates one of the techniques that Duane uses so effectively to relax his audience and augment understanding, that of humor. He also brings the mysteries of science into clear, comprehensible language, using analogies and diagrams, models, whatever it takes. A physics PhD, retired research and development specialist with Bell Laboratories, and current community college and elder hostel professor, Dr. Ray has applied his skills in the commercial world and taught in the academic one. Please welcome back Duane Ray.
And thank you all for the opportunity to talk about science. I think it's something that really needs more exposure in our society today because there's so many things that are relevant that need decisions with good scientific, logical background. Today is especially true, and although you don't read too much about relativity and quantum mechanics in the newspapers, you're going to read an awful lot about DNA evolution, and particularly some of the things we're going to talk about today, genetic engineering, cloning, stem cell research, and stuff like that. It's a broad topic, and I've only cherry-picked, of course, and I hope I have strung it together well enough that you'll be able to sort of get some sensibility out of the whole thing. One of the things I benefit from all of this is that it forces me to sit down and put things logically together, and also I go across so many different things. I uncover an awful lot of new things, and that's true today. I found out, for instance, that snowmen contemplate their origin, evolution. But the one that I really was surprised to find out about is that paper clips are the larvae of wire coat hangers. So, we're all learning together this morning. Let me give you my disclaimer. As Ziggy, whenever he goes to the top of the mountain, finds that, this is the facts of life. The meaning of life is 2 mountaintops over. We did that a couple of weeks ago, and we're not going to go there again. And I really like this description of science because I think it's very comprehensive, and it holds in all the situations that I've sort of seen. Science is neither a method nor a body of knowledge. It's a body of changing, learned opinion, aspiring to be true. Certain facts about nature and history, our grasp of those facts is constantly changing. And it's certainly true. And I think that fits and works for pretty much everything. Here's basically what we're going to talk today. We're going to talk about some DNA. We're going to talk a little bit about evolution. And then we're going to talk about some biological stuff, bits and pieces here and there, with genetic engineering, cloning, and stem cells kind of at the end after we have some background and do some different things. One of the things I think that's an important way to look at things is that a lot of things are very simple in the small, and the essence of life's complexity is not with the rules, but with their repeated use. So you take something simple and you do it again and again and again. Pretty soon you get pretty complicated. And we also find that whenever you have a lot of something, it becomes different. So you take a few nerve cells and the worm is able to crawl, you put enough of them, and you get intelligent thought. So let's start off with something. You're skeptical already. Let's start off with a very simple tree-shaped kind of thing here. And then we expand that, and it becomes a little more complicated, and it becomes a little more complicated, and it gets a little more complicated, and it becomes even more complicated, and we keep doing this until finally it becomes pretty much of a mess. If you try to start thinking about it at first, you might be kind of overwhelmed, but then you see that, well, it's really just broken down into individual pieces. Now let's lay one more complication on top of that. A model that Richard Dawkins presented in his Blind Watchmaker back in 1986. And what he wanted to do basically was have a computer model that he would be able to show how evolution might be able to convert into other types of forms and pictures and drawings. And he tried to make it so that it would be very similar to what we observe in evolution around us today. So first of all, because he wanted to make it sort of comprehensible and reasonable to us, he said it has to be symmetric. My right-hand side is very much like my left-hand side, so it's got to be symmetric that way. Simple branch sequence, like cell division. Simple changes, small changes at a time. The number of times a branch can change. A simple recipe is applied many times, and we'll see that in just a minute. And then he had 9 gene type operations, that is, things that could make slight modifications to branching mechanisms. And the angle of branch, number of branches, and 6 other characteristics. And here's sort of what they look like. So here's the basic tree. And if you apply this gene, it squirts it a little bit this way. They're all pretty much the basic tree, with slight modifications. Okay? So then if you start on a computer and you have first generation here, that simple branch. And then the computer puts in some random changes with these genes. And it comes out with a whole bunch of different things. And then you're sitting there, I choose that one. Like Mother Nature would choose something. Okay, so you're the person, you're Mother Nature making a decision. And you follow these things down, and you keep doing that, and you keep doing that, and you keep doing that. And after only 28 generations, you end up with a rather complex looking kind of thing. And as a matter of fact, if you decide you want to do that a whole bunch of times, you have different branches of evolution. And so you end up with just all kinds of stuff. Does that look like spider, frog, whatever? Well, here's sort of a summary of things that are reasonable after he did that kind of thing, after just a certain number of iterations. Swallowtail, man in a hat, cat's cradle. Well, okay, tree frog. Boy, does that look like a tree frog. Scorpion, a lamb, jumping spider. So the whole point, of course, is you take something very simple, you make slight modifications a long period of time, and there you are. So the question, well, so that's to illustrate, in fact, that evolution could be very readily carried out in that manner. One of the other things I want to point out is that a simple code can convey an awful lot of information. If we take a look at Morse code, and we're surrounded by them all the time, it's nothing new. You have A is represented by a dog. Whoops, backwards. B is da, did, did. And C is da, da, did. Now, the thing is, you see 2 dit and da, so that's 2 things, but really it's 3 things because there's a space between it, and then there's a space between letters, and then there's a space between words. Okay, so there's really more hidden in here than you might think. But on the other hand, if you have a binary code, we all know our computers run on these things. So these particular groups represent these numbers. And if you know how to read the code, you know how to do that. And of course, we're even more familiar with something like this. If you take the, T-H-N-E, and you add an M, totally different meaning. Theme, totally different meaning. Just slight variation gives you different things. Time, change a different part of the code. Okay, so you have a whole bunch of different things. And here, heme is buried in the middle, and it's part of the blood. So just by using a simple code of different letters, we're able to get some very complicated kinds of things. The code of life, we know, is written in DNA. You've probably all heard of that. And that was discovered back only in 1953, just over 50 years ago, 55 years ago or so, by Watson and Crick. And this is basically the scheme that they had. You can sort of see a winding of this helix around here. It's very well described in a book, the discovery of this in the double helix. Fascinating book to read, I think, a little bit controversial because it lets some pretty strong statements hang out about personalities and so on. If you really take a look at the helix, it's not that clear open space thing because atoms have size, and when you start jamming them together, you start ending up with a very complicated thing like this that's jammed together. But there are a couple of things about it that are kind of interesting. This is a helical spiral with 2 long chains tied together. So it looks sort of like a spiral of this sort. And really the basic code that's going to come is because you have T is always joined to A, and G is always joined to C. Could be C and G on different sides and so on, but you never see a G connected to a T. So the code is really combined in this spiral structure. So G is always combined with C. And the sequence, we'll talk about a little bit more, whether it's A, G, G, T, D, A, G, 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 C, A, that's a code sequence like reading in a book, and we'll get to that in a little minute. One of the things that's notable about this that's kind of interesting too, is that if you unwind these different pieces of the spiral, which can be done, and is done all the time, and then you start forming on those pieces that you have separated, you form 2 identical pieces up here, because you know that when you split this out, this A here, the old piece, you have to join it with a T, and this T you have to join it with an A. So by the time you zip those all together again, from stuff floating in the solution, now you have 2 pieces. And if you strip those apart and do it again, you get 4 pieces. And that's used, actually, we'll talk about that later, whenever they want to increase the amount of DNA that's there for criminal investigations and parody and things like that. Okay, whenever we take a look at how those molecules are actually put together, what they look like, with physical size, they're much more like this bunch of stuff. This is one rather simplistic way to look at it, sort of a stick between the bonds and then a small atom here, so you can sort of see through things. But real atoms combine in complicated ways to make really complex molecules that we're all made out of. These long-chain polymers, proteins, whether it's your skin or a piece of hair or your liver or pancreas or whatever, it's made up of a whole bunch of different types of molecules that look sort of like this. So the big thing is that they're very complicated. Shape is very important because you can't kind of put this one beside that one and fill in all the gaps unless it works a certain way. And the same with its interaction with the other things. And the big question is, well, how do you ever decide how to put one of these long things together? How do you take individual pieces like this and with only 20 types of building blocks assemble them into a long chain that's going to be this kind of complexity? That's exactly what you want to have. Well, DNA has the answer. And as a matter of fact, if this is a DNA helix, these are the different indicators inside that make the code. And if we take a little closer look, the codes for amino acid building blocks, that is, there are 20 building blocks, they're called amino acids, and we're going to take just those 20 building blocks, and we're going to put them together and make these long-chain polymers of all the different kinds of proteins that are in your body. Okay, so here's one code, GAC, that's in this DNA helix. That indicates a certain type of building block. It's like a certain word. Is it a noun? Is it a verb? It's a certain type of word. You're going to combine it together into sentences. Here's another one. Here's another code, TGT. And then here's another one. So all along here, there's a sequence of letters that tells you the code for a particular amino acid building block. So if I combine these amino acids together in the proper code, I'll get the proper protein that I want. So I end up with the function that I need to have for the body and the cell. Basically, this is sort of the way it goes, and I didn't want to go into more detail. It's interesting to look at, but it just gets too messy. And I'm kind of skipping over a few things. But the idea is that you have a DNA recipe by these A, G, C, T stretch out along the length of the DNA. And in higher animals, that DNA is always kept inside the nucleus of the cell. It never goes out into the cell itself. So that information about the recipe to make different proteins has to get out into the cell where the manufacturing machinery of the cell is. And what happens is that a message goes into the inside the nucleus to the DNA. It says, I need a certain type of protein. So the DNA of that part unwinds. It makes a similar RNA blueprint of that gene. And then the RNA, the messenger RNA, goes back out of the cell nucleus to the manufacturing part of the cell. And this is where the protein is made. It has access to all these 20 amino acid building blocks. And it makes all the different proteins that are needed. Here's one that's insulin. And this is actually what the sequence, if you could read those little bitty things, it tells you which amino acids in which order and how they're tied. This is actually the structure for the insulin, specified in one way. So that's sort of how it happens. Let's talk a little bit about scale also, because that's always something that is amazing to millions of people everywhere, I think. If, for instance, this BB in the hand is an atom, then a marble would be a simple molecule. Then, if we compare that size, and we have something the size of a cat, would be the equivalent of one of these complicated molecules. And then, if we compare that to something bigger, this might be a cell, this whole ship. Here they are, see now, here they are here on the dock. And this tractor trailer would be some kind of molecular structure inside, like a mitochondria or something inside a cell. We can talk more about that. Matter of fact, we will talk more about that. So if I take this then, and this is our cell, and I expand that, then this might be your liver, or your stomach, or your heart or something. So here's a cell, and then this whole mountain range area would be an organ within that. And then that mountain range would make up the whole multicellular organism. So this little bitty thing down here with a mountain range, and now this is the whole organism. That's all of me. And here's my liver, and here's my heart, and here's my, whatever. So that's the kind of scale. So you go from a BB-sized thing from an atom to all of North America for a real individual. That's sort of the way things go. If we take a very simple cell called a prokaryote, one of the very earliest kinds of things that existed, it really is very straightforward and simple. It has a membrane around it, holding in this bag of stuff, so that it's not diluted and disappear to the surroundings. This is a DNA strand, just kind of hanging loose in there, and very accessible. It's very simple, very short relatively, and so it's very accessible to manufacturing processes that are needed in here. And the ribosomes carry out a lot of that, and we won't go into those details either. But we'll talk about some of the pieces inside when we get to other more complicated cells. These things already are able to do some complicated things that have turned out to be very useful to know about, because they really help a lot in genetic engineering and understand how antibiotic resistance develops and so on. Bacteria don't need to divide in order to be able to pass on from information that they have. As a matter of fact, if this particular guy spent his time in a hospital and picked up some immunities to certain kinds of antibiotics or whatever, he's able to say, here's one, he doesn't have it, so he's able to make a connection with this other bacteria and squirt some of that information across, okay, and then they go on their happy way, and each one of them now has that ability to be resistant to antibiotics or whatever. So that's one of the reasons why things happen very quickly, to build up immunity if you allow these things to exist. And as a matter of fact, here's a picture of one such bridge of these bacteria sharing information that way, genetic information. So the genetic information, or the information about building proteins in the cell, is not just in the DNA, but all these little DNA rings also are added. And actually these would be shared also whenever it would divide, I'm pretty sure. And it can't necessarily just display this combination of transfer with other things like itself. It can also share with plants. Here it's sharing with yeast. Here a virus, a virus is a small particle that really doesn't have its own cell mechanisms particularly in it. It doesn't do much. All it has is DNA that's able to, and it's able to attach itself to some cell, squirt its own DNA in, which knows how to be a robber baron, take over all the chemistry of the cell, and start reproducing itself to the detriment of the cell. And the cell kind of just dies, squirts, and all these virus particles that have been generated by the cell's mechanisms now disperse and start infecting all the cells around it. Okay, so those kinds of things happen all the time. And that means now you have this mix and match all over. And so if you have a little fly here, and a mite's on that, and he sucks out some of the genetic information here and carries it over to some other fly and spreads it out. There's just all kinds of mechanisms. Simple things one at a time, just more and more and more complexity, and pretty soon you have a world. Here's another thing that I thought I would talk about, because I think we should understand that higher animals, like humans, are really rather more complicated also in the DNA. The DNA inside each one of those cells that you have in your body is about 3 feet long. Okay, it's very thin, and it's all wrapped up very tightly, and we'll see how in a minute, sort of. And it's got to be really carefully wrapped in order to be inside that little bitty cell that you saw on that wharf beside the big ship. But on the other hand, it's got to be possible, it's got to be organized such that something can go in, unravel the appropriate part, find out the gene, that is the recipe, that it needs for the particular polymers that it needs to make, whether it's insulin or something else, in the cell. Now, it ain't that simple, really. Because we used to think it was, but it's not. Because all the information about a gene is not all right beside itself along the DNA. It's kind of scattered. And sometimes it's scattered between other pieces and between things that don't mean anything. So you might have the messenger RNA for protein 1 is from this piece, this piece, this piece, and okay, they combine here, and something has to be chemically able to be smart enough to recognize these different things, stitch them all together, and then start making the protein. Remember, we had millions and millions and millions of years to learn how to do this. And we built upon things that were already there. Just like in that model that Dawkins had, you build on what was there before to make something more complicated. So this is basically the kind of thing that that happens inside human cells. Okay, here's human DNA package. It's packaged into 23 pairs of chromosomes. So we have a total of 46 chromosomes. That's no magic number or anything. It just happens to be the way things are packaged. You start off with DNA helix like this. You wrap it carefully on specific sized chunks of material. And then you organize those very carefully. Then you wind those up in a different way. And then you wind the windings up in a different way. And you come out with a thing that looks sort of like this. And we all have very similar things to that. We all wind them in the same way. So if you take somebody's nucleus from a cell and you open that nucleus up and you spread them out, you'll find a whole splay of these chromosomes. And the way they identify them is really by the size of them. That's what they did originally, and we continue to do that. So that you start off with this is number 1. This is a big one. And then number 2 is a slightly smaller one. Number 3 is a little bit smaller. So you lay them all out that way, and you end up with 23, and then they're duplicated because mother, father come in and combine with those kinds of things. So this is the kind of structure that you end up with. Very carefully organized, very complicated. One of the things that makes this a little more plausible to understand how does all this stuff happen, doesn't take forever for this to occur, is that molecular interactions happen a million times a second. So, things happen, and that's just it. I mean, whenever I wave my arm out there, all kinds of muscles and energy and kinds of things happen, and it all had to happen very quickly. And compared to a million times a second, okay, that makes it a little more plausible. Not understandable, but plausible. This is a fellow who helped complete the laying out of the details of the human DNA genome. And of course, there's always a little bit of a controversy about that because they try to keep, whenever the government started doing this, and the government started down a rather methodological approach to this, and it was rather slow. It was taking several years. And then he came in with a really neat idea, a totally different mechanism. And he said, gee, I can do it in 2 years starting from scratch or something like that. And so he got together with the government and they kind of cooperated, funding and techniques and so on. And so it speeded up the program dramatically. Now originally they said, well, gee, we really shouldn't do just one person's genome because then that would almost be an invasion of privacy and people would be able to look at that and tell different things and so on. So we don't want to do that. So what they did was they took DNA from several different people and they just kind of mixed it all together. Now, it turns out that that's not a problem because people know how to cut the DNA at a particular type of place, and they all have similar types of places like that. So they could take this from one person, this sequence after this, and from another person for the sequence before that, and so on. So they could combine this and not get it all in a big jumble. But then he said, well, really, it's useful also to have an individual's DNA, and I think we'd learn a lot from that, so I'll put mine out there. And now Watson, the guy who helped discover DNA, has put his genome out there. So it's all specified, all laid out. And this guy actually was here and talked in town not too long ago, I think maybe last year it was. And it was kind of interesting. He's published his DNA. You can go on the internet and pull it off. It looks something like this. The specifications of it. Here's one page, and this little bit is this. And of course, each one of these little things gets blown up. So there's a lot of stuff there. But you can get this, I think, on DVDs even if you really want to go into it and use it for your research or something like that. Maybe we can get a discount for you. The other thing, well, one of the things that he made a comment about whenever he saw that was, you sort of know, we pretty much have a pretty good idea about which genes influence, for instance, eye color. There might be a half a dozen that contribute to that, or maybe more, I don't know the details. And he said, now looking at my genome, knowing that these genes are involved and what they do and so on, there's no way I could guess that my eye color was going to be blue. So we have an awful lot more to learn. We've just started scratching the surface, but we're making tremendous progress. We're really moving down the line. Here's an old estimate of how many genes, that is, how many recipes that humans have in them. Now it's about 30,000. Now it's sometimes hard to sort that stuff out. Remember, some are intermixed and so on, so it gets to be kind of tricky to do that. And so now they have different numbers, and so the roundworm has 20,000, and the fruit fly has 13,000. So what? It doesn't really mean a lot. But what it means is that the fruit fly is making a lot of the same kind of proteins for its existence that we make for our existence. And this also is an indication of, we're all in this together, we're all from the same stuff. One of the other comments I'd make also that I didn't mention earlier, I think, is that this DNA, all that coding, it's the same for everything. We're talking trees, fish, microbes. It's all the same code. It's all the same stuff. DNA is very much the same. So it's likely we came from a common origin, and there are plausible reasons to understand why that might be true, but I won't go into much of that right now. I'll mention a little bit more later. The genetic similarity, that is, the number of recipes that are similar, among human individuals, 99.9%. Ventnor, I think, has come out more recently and said he doesn't think it's quite that high. There seemed to be more variation among individuals than was anticipated. The chimpanzee, 99%. 70% mouse, 60% fruit fly. So there's a lot of similarity in the kind of molecules that we use in our everyday life. Here's an example. Just cherry pick this one particularly. If we take a look at hemoglobin, and a lot of mammals have hemoglobin, because that's what gets the oxygen around to the different cells. If you take a look at the total number of these building blocks that there are in hemoglobin, and they're different amino acids sequenced in different ways, and we have about 287 total amino acids making this complicated hemoglobin molecule that clumps the oxygen, carries it out, and then gives it off to the cells where it's needed. And we take a look at Homo sapien and chimpanzee. Every single one of those building blocks is sequenced in exactly the same way. If we take a look at gorilla, in all of these 287, only one is different from us. If we take a look at a gibbon, only 2 are different. Now, which do you think is the closest relative to us? Now, of course, just looking at hemoglobin, that's just one thing. But it's indicative, and it makes you go and look for some of the other things. So chances are chimpanzees are pretty close to us, and gorillas are not far behind. Okay, squirrel monkeys are a little further, rhesus monkeys a little further away, but not too far. It's only that many out of a couple of hundred. And you can see that again and again and again. So, it's really kind of interesting to see how that stuff goes. I mentioned how we go in and we maybe go to a certain part of the DNA in order to be able to break it off. And I said, maybe humans, you're going to look at it from one person on this side and another person on that side. Well, out of these millions of sequence building blocks, how do you know this particular one? Well, let me put it in something that's a little more everyday. If we have a form, so this is form number blah, 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 from some government or agency or whatever, okay? And we've all seen those kinds of forms. And we also sort of know that then the name, last name probably is going to be up upper left-hand corner. Signatures are going to be on the last page, right, with the date. I mean, we know about those kinds of things. And as a matter of fact, if we put things in, we could expect that last name is not going to change real frequently. The first name is going to change more frequently because he's married to somebody who's got a couple of kids. Okay, so that changed a little more. Portland is maybe not going to change too much. The zip code is not going to change as much. Oregon is certainly changing less. This zip code is changing more. Address is going to change more. And at this address, there might be several people. So you can also tell the frequency with which these things might change. There's also fine print down here that never changes. So we don't want to look at that if we're looking for differences in individuals and people and so on. We know that if, when we go to this particular form, some particular group of sequence in the DNA, then we'll follow that, and we know that certain things are going to change faster than others and what to look for. Phone numbers, all that kind of stuff. So we know where to go, what to look for, and what to expect in a general kind of sense without knowing the real details. And that brings me to forensic analysis and how we understand you can capture people using the DNA or identifying relatively people compared to other people. You can distinguish them. Basically what they do is they get DNA from the nucleus of a cell, and that DNA may be laying around at a crime scene, or it may be a paternity test, it may be from cells taken from swabbed from inside your cheek or something. Anyway, you get those cells, and then you take the nuclei out of those, and you start looking at the DNA, and you go into certain areas, and you split the DNA up, and then you take that, and you increase the number of DNA molecules you have by multiplication techniques that we know very well how to do. And so then you take a lot of these, and some of the pieces are long, and some of the pieces are short, and we start laying those things out. And we put a gel here, we put an electrical potential across, and these pieces of DNA are charged, so they start moving through this gel. And the short ones aren't blocked as much by the gel, so they move faster. And the bigger ones are blocked more by the gel, so they move more slowly. So after a day or whatever, several hours, pretty soon you have an array of the long ones and the short ones, and they pile up in regular quantities there. So you can tell how many short ones, how many long ones different people had, and you can quantify that. You break these things up, you put radioactive materials on them so you can get a radiograph from them, take a photograph basically. There are other techniques too, but that's basically the type of thing that happens. And then you end up with a sequence that looks like this. Okay, and you have controls and so on. So basically if we have a crime, there's a sexual assault, then if this is the female that was involved, this is suspect 1, suspect 2, and here's the sperm DNA that was found as evidence, and here's the boyfriend. So you're checking all of these different kinds of things, and here's the sperm DNA that was in the sexual assault, and we take a look at this suspect 2, and that doesn't match up very well, but suspect 1 certainly does. And of course the boyfriend over here, he doesn't match up at all, so it definitely was not him. So by using these DNA things, you can very easily zero in and find out who was the person associated with just a very small amount of stuff that you collect at the scene. And as a matter of fact, I read recently that they are now able to take something like 6 or 8 cells, incredibly small amount of material, and amplify that well enough using some polymerase techniques to be able to give you enough DNA to start doing this kind of thing. That has really been a benefit in a lot of places. And as a matter of fact, this Innocence Project has been very active for the past many years. And the last I heard, there were about 200 people that have been released from prison because of that. Something like 9 of them were on death row because of murders and so on. So you can just imagine why people are concerned about the death penalty. And this Innocence Project tries to make sure that they get to those people before it's too late. I think this has also motivated a lot of different states to get rid of the death penalty. I don't know whether it's still there in Texas or not, but I think they still have it. Well, I sure hope they do a lot of DNA, but see, that's only part of it. One of the things that comes out of this is first-hand eyewitnesses are very inaccurate. Because time and again, they have face-to-face confrontations, and this eyewitness identified the person. Absolutely no mistake about it. And DNA showed it was not right. And any of you that listen or see any of these, read about some of these psychological studies and so on, but that's another whole story. But the validity of eyewitness observations is just totally trash. So we really need to get more of this involved. The other thing that kind of stood out to me was that only the states in yellow are the ones that have a policy of compensating the exonerated people. So the other people, you blew 20 years of your life, tough. We'll let you out, but you're on your own. Now they might be able to come back legally and do something, I don't know, but there's nothing that automatically gives them, I'm not sure how much. One of the things that's interesting is that you can go on the web, and for $99, if you send in cheek swabs from people, you can do a DNA analysis and find out who the father is, or are we really related. And one of the elder hospital things I taught, there was actually a doctor there that had come from someplace down, I don't know, Georgia or someplace, and he lived in a town where some maybe cousin had the same name. And they thought, well, they may be sort of related back, whatever. So they paid to have their DNA analyzed to find out whether or not they were in fact really cousins. And it turned out that, yeah, they were, but it was pretty far removed, but they were able to analyze all that kind of stuff. So there's a lot of interesting things that go on with that. It turns out also that the $99 is only if you have the standard cheek swab, but if you're really picking it up off somebody's toothbrush or cigarette butt, that costs an extra $75. Well, it requires a little more work. I mean, you've got to get that. So, but what it means is that a lot of paternity choices. One of the things that amazed me when I was reading more about this is that 1 out of 7 has the wrong father. 1 out of 7. 14%. That's 5.
Yeah, monogamy really works. Selection bias.
I was right there, but I don't remember a thing. Okay, if we take a look at paternity, let's move along to a slightly different effect, but we can get so much information out of DNA analysis and information. Let's take a look. If we had a parent back here, and the descendants, these are different generations, okay, and they move down to the present. Some don't make it all the way, some lines stop, okay, who knows for why, but some of them make them to the present, and you can see there's a line to follow back, and that's kind of interesting sometimes to follow. But if you have somebody up here, you can follow the female line back, and what we find is that in Africa, about 140,000 years ago, there was sort of a common group of women that were, the mother of mankind that we are involved with. And the male line goes back about 60,000 years. They're able to trace that to some common sort of theme. A couple of things here. This is the Y chromosome for the male line. That's only a very specific thing. That's kind of harder to get hold of. So this analysis came a lot later than the female line. One of the things that's interesting about the female line is they don't really go with the female DNA. What they go with is the DNA of the mitochondria that's in the female cell. We'll talk a little bit more about that later, but basically inside the human cell, there are mitochondria, which probably crawled in early on in evolution and had a symbiotic relationship. And that mitochondria, which does a lot of very useful things, there's a lot of mitochondria in the animal cell, has its own DNA. And that DNA is what they were able to find, and they knew that that was from women, because when the egg is made, the sperm doesn't bring any DNA from that mitochondria. He only brings DNA that's going to go and unite with the nucleus to make the individual. So any of the mitochondria DNA is uniquely to the mother. So you know that it came from the mother all the way back. Okay, so that's one of the things that they do. And by using that kind of analysis, and this is more recent stuff than the early stuff, they were able to know that about 200,000 years ago is roughly when mankind started. We know it started in Africa, and then it moved out, and they were able to get times just from the DNA of how it moved to different parts of the world. Now, one of the things that sort of goes into this and some other analysis is, for instance, if you have some organism up here and it's making DNA, okay, and time goes on and it has some kind of a blurb, a dislocation, and then it comes down here and then it splits, and this branch goes over to this species and this branch goes up. Now they both have that dislocation, okay? So they carry that along. Now if this one gets another dislocation, this one doesn't have it. So if I find both of those two dislocations on this side, I know it's not related to this at all because they branched up here. If I find this location on both of them, I know that they have a common point up here. Okay, so by doing some of those kinds of games, you can get together and find an awful lot of information and lay out some of the time frames. And, you know, it's never quite as simple as that, but there's a lot of information that can be gained. The T-Rex is you know, there's a lot of stuff that they have found, and it turns out that they were able to find amino acid chains. These are the building blocks, and they were able to find DNA sequences from T-Rex that were still viable, that they could go in and analyze. And there was always a couple of questions about dinosaurs. You know, were they really related from birds? Are they really lizards? Are they, you know, whatever? And it turns out by using those sequences, they found that 3 sequences matched collagen for chicken, 1 sequence for a frog, and another sequence for a salamander. Now you say, well, you know, 3 sequences, oh, come on, you gotta be kidding. Well, on the other hand, wouldn't you know that those 10 sequences say German, and this says United States, and this says Mexico? So it's maybe not so far off that it's plausible that, you know, scientists know what they're doing. A few Snickers over here. Here's another thing where I mentioned to you before about the fact that we had a symbiotic relationship develop. Perhaps a long period of time ago, we ended up having some small thing that turned out to be a mitochondria. Mitochondria produce the cell energy molecules that are basically batteries that then get sent around in different parts of the body to provide energy for local chemical reactions that are needed, that need some extra energy. We don't get it from the sun, we get it from chemicals induced in our food and so on, and then it gets packaged in these little things that, like batteries, okay, they're very standardized, and they go around and help form all of these different chemical reactions to cause them to take place. And the idea is that this crawled in and got a symbiotic relationship with this, and actually Lynn Margolis was the person who was pushing this back in the early 70s or 80s, I forget which. That's the first wife of Carl Sagan. And she was poo-pooed a lot. And it turns out that, in fact, she was absolutely right. So that's carried on and has actually been proven to be pretty convincingly true now with a lot of the DNA studies and so on that have been going on. This is how complicated an animal cell is. Here's the nucleus. Here are some of these tractor-trailer kind of trucks outside. Here's some mitochondria somewhere, and there's other mitochondria, lots of mitochondria around here, and all these other things. They're sort of like organs within the cell. It's not a simple just bag of stuff. It's got a lot of things going on, and to get certain functions to carry out, you go to certain parts of the cell. So it's very, very complicated. Here's a plant cell. Here's an animal cell. They're both pretty complicated. So by the time we've gotten to this point and we're turning out trees that do wonderful things and have beautiful colors and so on, it really requires an awful lot of complexity in the DNA and then the organizing of that and then the carrying out of all these different functions that are necessary for those things to survive. One of the things that was really dramatic and just blew everybody away back in 1987, a deep-sea submersible decided that they were going to go see what was on the bottom of the ocean down near the Galapagos, I think is where they started. And so they went down, and they didn't know what they'd find, maybe some fish, maybe some crabs crawling on the bottom, whatever. Of course, down there it's so dark and the pressure is so high, there's no light to support anything. What can grow? Well, they turned out, this is not the particular one, but they found these smokers, which are basically volcanic-fed things, that is the heat of the volcanic part of the earth really hot, heats up this seawater, dissolves a lot of different chemical minerals that are there, and blows it out with a lot of particulates and causes what are called these smokers. And they found that there were all of these kinds of worms. These are long worms, and these are clams and other life around them. And a closer look at these things, there is no life. There's no light down there. All of these things live because they're using different kinds of chemistry for their metabolism, to be able to survive and to produce and grow and so on. And then other things feed off of the different materials that are grown, biological materials that are down there also. So there's a lot of stuff down there. And of course, now that they've looked, it's been 30 years, and they've found these all over the world in some of these deep sea rifts where the plates are coming apart and you get all of this upwelling. Well, whenever they found these, people were doing analysis of DNA for bacteria and so on, and they found that what they found there didn't fit in with plants or animals or some of these other bacteria that they were aware of. So it caused a whole new view of things. And as a matter of fact, it came up with this whole area here called archaea. So we had the bacteria, which were the early things, and all this stuff came from some primordial whatever that we don't know much about. And then some developed in bacteria, some developed into multicellular things, eukaryotes, and here's where you have the animals, and you have fungi, and you have the plants, and so on. And then this whole new thing came up with the archaea. And we've only known that for the last 30 years, 20 years, 25 years. Here's another thing I wanted to point out. Lynn Margolis is the one that was pushing about this, where symbiotically mitochondria went over and joined. Also, a lot of the photosynthetic things have a symbiotic relationship with various plants and animals. This was in the 1980s, okay? I couldn't remember whether it was 1980s or 70s. But she's the one that pointed this out and then brought it really to fruition. Let's talk a little bit about evolution now. We really didn't talk too much about that, but let's do that. Is it reading 10? No, it's 10:30, okay. Still have some time. Evolution basically operates like this. Population tends to increase indefinitely. As long as there's food and so on, it'll keep reproducing and growing. In a natural environment, however, population numbers stabilize at some point. You run out of food, or there's not enough space, or whatever. Things start to eat you. There must be a struggle for existence, since not all organisms produced can survive. There's a limit on food or light or whatever, and the ones that are best adapted to taking advantage of that are the ones that survive. There's a small variation in every species, and in the struggle for existence, those individuals with a variation better adapted to the environment survive and leave behind more offspring than those less well adapted. That's sort of basically Darwin's view of natural selection and how evolution changes and adapts as life goes forward. The variations are small, but cumulative. Suppose only 1/100% of the population has this variation, or even less than that, whatever the number is. Think of compound interest. The next generation, there's a little bit more. The next generation, a little bit more. And the thing is, we're usually thinking in terms of 10 or 20, 30 generations. Before my grandfather, I don't really know anybody in my family, but we're talking about 10,000 generations maybe, or 100,000 generations, way back. So these accumulate like compound interest and can have a big impact on what actually results. Yeah. This is the evolution of the cat. I'd like to point out it crawled out here, became a ravishing cougar or something like that. And then it found that it could sucker some humans into getting I mention this just quickly because I think it's necessary to include at least some fossil stuff in a talk like this about evolution. This is actually the Burgess Shale, which is up in Canada, and I've been up there, and you're limited. You can't just go yourself. You have to go with a guide, with a park ranger, and they'll get like 20 people or something. You pay a fee, and they hike you back in, and then you can see some neat trilobite fossils like this and a few other things. And the really nice ones they have locked up in a big metal box that they take out and show you when you're there and so on. It's just interesting to see the area. The area of the Burgess Shales is right underneath here someplace. So it's kind of fun to do and interesting. I forget one of the things I was going to make a comment about also. But at that time, the kinds of things that were observed were just totally new. And the book by Stephen Jay Gould, Wonderful Life, describes this in great detail and gives you a lot of interesting insights to that. Stephen Jay Gould, by the way, was in 2001 was the Humanist of the Year. And there's some others, like Wilson, who was also a humanist. I forget what year he was, but in the book that you have, I point out some of those things. Intelligent design is sort of a quick follow-on to that. Oh, the other thing I wanted to mention about the fossil thing. Let me go back just to that. You have fossils like this. They don't show up too often. They're kind of rare to find these. And one of the big things that the creationists and stuff were always finding was that, well, you know, you say the dinosaur is from a bird, but where's the intermediate step? And so that was always a bit of a problem. Now with DNA, that's not nearly as much of a problem, but it was a real issue for a while. And I think if you reflect on that, I once saw where if all the people in the United States, that's 300 million people, each of us with 205 or 6 bones, if we all died right now, then somebody came around 200 million years from now, how many bones would they likely find? It turns out it would be about 54. Because, you know, some are eaten, some just rot away, some get flushed out, you know, whatever. But they disappear. So the fact that you don't find some of these intermediate things isn't surprising at all. And that's why this was a dramatically important thing. And as a matter of fact, they languished in the Smithsonian cabinets for a long period of time. They were first found in 1909. And he misrepresented what they were and put them into different classes and phyla and so on, species. He was the head of the Smithsonian at the time, had a lot of business to take care of and things to do, so he probably didn't put as much attention to it. And he didn't have as much the tools at his hands that we have now. So he mislabeled everything. He just kind of stuffed them into previous categories until some graduate students, I think in the early 70s or 80s, came in. And one of the marvelous things about the things found here is that a lot of them were soft animals, and it's because that things slumped down over and buried them in a way that they couldn't decay. It was anaerobic kind of situation. And one of the things that this graduate student realized was that they're just smushed flat and everything is still there. So if you start picking off the layers with a dental pick or something, you could go down and look at the lungs and the heart and the liver and the legs underneath. All the stuff is there. And that's why you're able to come up with pictures like this, showing these really weird and strange creatures that we don't see anything like anymore. But that's also where they see one of the first worms that's a notochord at the first spinal column, and we're all descended from one that we saw there. Okay, let's go on to intelligent design, because I think that also that's sort of the wedge that people sometimes try to do to get away from the idea of confronting evolution. ID looks for intelligence of design, evidence of intelligence. One evidence of intelligent design is irreducible complexity. Remember I mentioned Behe was big on that with his flagellum and the bacteria and so on. And they would say there's no possible way for the function to have evolved in a step-by-step manner. Well, one of the things that's fairly simple and well documented to talk about is in fact this. The human eye and the eagle eye, and as a matter of fact, even the pinhole eye of the nautilus, are really pretty miraculous. So let's go back and see how you might expand your view of how evolution could possibly have produced these things. Because, you know, an eye, unless it has a function, the idea of intelligent design is, you know, if it's in a less useful form previous to this, there's no reason it would evolve to provide this more useful form. And let's take a look at how many different types and styles of eyes and what benefit you might have from it. For instance, if you had a pinhole, you can kind of make out an A here. And if you have a water bag, you can make it out a little more clear. It's sort of a lens type thing hanging in that plastic water bag. And even a wine glass will work. So, you know, it doesn't require an awful lot of fidelity to give you a benefit about what's there. If you're trying to read the treasure map, that's close enough, and it'll work. Notice also now, if we have, and even simple bacteria, by the way, have a sense of light, okay? So they can move toward light or away from light, depending on what they want to do. But even simple bacteria have an ability to sense light and move in a direction toward or away. So there's a pretty basic level at which we have those proteins that are susceptible and react to light. Now, if, for instance, you have a little cup like this guy knows the light is coming from this direction because these are in shadow and these are not. And so it gives you sort of an indication. And you can amplify that a little bit into a cup, and these are actual eyes from creatures that are out there. And here's another one where you have all these types of slots that show. And if I put a pinhole in front of that, it gets rid of a lot of this garbling, and this pinhole just forms the image along here. Much better image, maybe not perfect, but much better. And here's just some other types of eyes, and I'll show you some more here in a little bit. I want to take a look at this one a little bit. Notice these are just long tubes with a sensitive area in the back, so it's focused along a certain direction. That's what helps it focus. And as a matter of fact, if I turn these around the other way, then I can get a pretty decent image of what this is. And now I was fortunate to be able to get some nice photos from Jim Johnson, who you see here, and take a look at some of these eyes from dragonflies, and they are incredible. So now we take a look at this, and there are 30,000 lenses. And if we had a 360-degree field, and we divided those up equally, it means that about 4 inches away, you resolve a little square, about 2 millimeters square. And of course, they might overlap a little bit, so you can distinguish more than that. You turn your head a little bit like this, and you can zero in on exactly where it is, because you know where it goes from one field to the other. And so these are really marked. Look at all the hairs that are on here. Great pictures. And here, 30,000 lenses. I mean, you know, it's just a simple eye. It's just a bunch of tubes collected, and you know, a simple number of tubes before, and now it becomes more complicated and more complicated because it's a definite advantage. And here, a limpet has this sort of general cup type thing. It gets a little bit more with this shell. The chambered nautilus actually has a bit more of a pinpoint pinhole up here. And then we have this kind of a thing, and then a common octopus gets pretty close to our own eye, and they're supposed to have very good vision. And this is a guy that, he's just so beautiful. But all these blue spots, the blue-eyed skunk, all these blue spots are eyes. And of course they're looking in all different directions. And they actually are kind of different. They have a mirror on the back, so the light goes through the retina. It has a reflective part in the back, and then it comes back up and is detected. Kind of crude, but it can tell light and shadow. And I've seen pictures, Nova or someplace, where the thing that really gets these, of course, is starfish, because they'll get there and pop them open. You bring a starfish within 3 feet of one of these guys, it's amazing how fast they can move. I didn't realize that. Really wonderful. And this is, you know, we have, in addition to the general change and evolution, every once in a while there's a really big hit that creates disaster for many, many species. About 250 million years ago, 95% of the species just disappeared from the surface of the Earth. 65 million years, they say there was a big meteorite that hit down off the northern part of Yucatan Peninsula, just off the coast, caused a tremendous explosion, pretty much caused maybe a long-term winter type thing because all the dust in the air and so on. And they think that that's probably what caused the dinosaurs to become distinct. It's clearly happened about 65 million years ago, and that's the most likely scenario with a combination of things that have happened. I personally have seen some other things, some alternative theories, and I think this is the real reason why I'm here. I found that in the literature too. I read a lot of that kind of stuff. One of the big questions that's always brought, of course, is is all this evolutionary process aimed toward producing something like us, as human beings that we are. We're the top, okay? And I think Gould and a whole bunch of other people generally say, no, that's not true. It isn't that we are headed towards something in some kind of random way that is directed in some specific direction. It's really a whole random kind of thing, and because you have a long period of time, some of those random variations get changed out to here, and then you have random variations on top of that, and slowly things, you don't get below zero, but you just, over time, move further and further away from the central part. And as a matter of fact, coin flipping is a pretty good example of that kind of thing. You know, you flip coins and it's random. Is it going to be heads or tails? But once you have a run of 15, that's pretty organized, right? Just random that you got to 15 doesn't happen very often. And the next coin you flip could make it less or more. So that's random too. But once I've got to that 15, I have the option of maybe getting to 16, maybe getting to 17. Okay, so that's basically the kind of thing that's involved with this. And so you have complexity with variation, random variation, and slowly at each point out here you have that variation, but it will tend to move in this direction. So you have bacteria and they get more complex bacteria, and as time goes on you end up with more and more complexity, and out of here the very, very small tail of things we end up with humans. And it's just all random variations, evolution. Sounds reasonable. And the other thing is, it's not over yet. I think that's something that we need to keep in perspective also. You know, we're here, and I think this guy, this chimpanzee here, he's pretty close to us, but I don't think there's any way that he could possibly envision what the next version was going to be of a aware, thinking person. So I'm wondering whether we are going to be able to envision what this next guy is. I don't know. I don't think we've reached the end. We've got another few billion years to go before our sun blows up, so there's a long time available. And then, of course, there are always things against us, though. Okay, I'll admit they're kind of cute, but I still say their herds need to be thinned. Have you heard that before? Let me show you another thing that I think is relevant to all of these kinds of things. We talk about DNA, and now we're going to start moving over into some of the genetic engineering and all this kind of other stuff. Other influences besides DNA that have some impact on things. So if we talk about a fertilized egg, I think the thing that you need to realize here is that an egg is not uniform in all ways. I mean, even a chicken egg, when you open it for breakfast, okay, it's not the same one side to the other. You have, you know, you can recognize different parts. Well, this is even more complicated. So a fertilized egg is going to have more of some kind of chemical up here than it has down here. So it has sort of a one end compared to another end because of chemical variations within the egg itself. Okay, so then whenever that divides, if it divides this way, that continues. So now, for instance, if I happen to be separating these, I could have identical twins. That would work. Okay, they have the same material, they have the same chemical distribution among the different parts of the cell. So I could have something like this happen. But now, if I divide this way, now I can't do that anymore. I can't take this piece here and this piece here and create two identical things. As a matter of fact, if I separate them this way, they probably wouldn't work because, generally speaking, we're going to need all the things up here to make things work properly for the long term. Now, each one of these has a complete set of DNA. So it's not like there isn't the right DNA there. It's that the chemical surrounding it is different. And that really has a big impact on future things. And as a matter of fact, the gene development, this is a fly larva, and they've started studying what are called host genes. And these have to do with the way things divide up and differentiate into the higher pieces of the animal as things go on. And these are individual cells, and you can see these are stained so that actually they're different already. And it's because the chemistry between the cells has an effect on things, and the Hox genes have divided out vertically so that they react differently to those chemical variations, and different genes are turned on up here, then are turned on here, then are turned on here, then are turned on here, so that chemistry has an impact already at this point. Okay, so now we start to differentiate out into more segments, and it just keeps going and keeps going. And so this kind of thing is sort of an indication of what's going on. They're so different, but they're so similar. They're both symmetrical. Whether you're talking a butterfly or you're talking a beetle, they have similar numbers of legs. Okay, this guy has legs instead of wings, but so what? I mean, they're not the same, but they're not that different either. Identical twins. So this shows a little bit of the variation that you can get from nurture as well as nature. You read an awful lot about, at least I have seen recently in the past couple of years, how identical twins are so similar. You know, ones that were separated at birth, they're great ones to study because you really see how much nature and nurture varies. And you know, the guy in California, he's a fireman, and he's done that all his life. And gee, the guy that was his twin in Florida, he's a fireman too. They had similar interests, but I mean, you can find all kinds of things like that, and there's a lot of similarity. But there's also some variation and impact on the nurture effect of things. So let's talk a little bit more about that. This is called epigenetics, an array of chemical markers and switches that lie along the length of the double helix are called the epigenome. Okay, and the epigenetics is a study of modification of the activity of genes without changing the gene structure. In other words, the DNA is still laid out with the same code. The genes are still there that give the recipes for all of these different proteins that you need in the cell, but sometimes they're activated and sometimes they're not activated, depending upon other chemistry that's involved. Now you have to choose the chemistry very carefully to make that change work, but they have, as a matter of fact, managed to do that. A groundbreaking experiment in 2000, fat yellow mice were given a diet loaded with chemicals found in onions, garlic, and beets that turned off a gene switch to give slender gray mice offspring. What you eat or smoke today can affect your offspring for generations. And as a matter of fact, typically all that kind of stuff gets reset whenever there's a generation change, okay? So that it goes back to the basic DNA, what's in those genes, how does that make all things work, and this other stuff that's overlaid because of your lifestyle and what you eat and so on kind of disappears because you're back to basics with DNA and coding. But that's not completely so. And as a matter of fact, they found that a change in nematode worms can be inherited for 80 generations. I think these are the mice. This is the mouse that was fed those chemicals from beets and onions and so on. And this is the offspring. And I think this offspring variation held for several generations. I don't know how many. I didn't see it in the article, but I think it was at least a half a dozen to a dozen generations at least. Maybe more than that. I'm not sure. Okay, let's talk a little bit about some modern miracles. Genetic engineering, cloning, and stem cells. These are all topics you read more and more about all the time. And of course, the big question about science is, are you a good genie or an evil genie? And that's always been the question. And the answer has always been yes. You know, how do we use it? That's what it really comes down to. And so genetic engineering, all of life uses the same DNA code. And that's true, as I mentioned, whether it's bacteria, trees, fish, whatever, that's both good news and bad news. It means that it's like having spare parts. You can mix and match and swap. So, you know, the ball bearing on your car, if it's the right size, can also go into your son's tricycle or your daughter's tricycle or whatever. It's a ball bearing. If it's the right size, you can move it anywhere you want. And that's basically what's happened. This is one of the first uses of genetic engineering. They had a daughter who had a problem that needed a genetic modification in order to cure it. And there was no way at the time to do it without a bone marrow transplant, and that was not likely to happen because of the mismatch and so on. So they decided to have a baby that would have the proper thing in order to be able to save this daughter's life. So they made a whole bunch of eggs, fertilized eggs, and then they selected from those eggs which would be one that would be appropriate. And that's the one that they brought to term, and here he is. So that's kind of indirect, that's just kind of sorting out among things. Mother Nature does this a little bit more directed than Mother Nature, but not that far. The genetic engineering, you can modify bacteria to produce drugs, like human insulin. It used to be that they would go to slaughterhouses and get insulin from cows or pigs or something, but a lot of people would react to that. So if you can have really human insulin, insulin is pretty much the same, but there's some variations. So if you can make it so that it's really human insulin, so they take the genes from humans, stick it in bacteria, and then you can produce as much of that as you want. Modify plants to be resistant to herbicide. Modify animals to produce special proteins, spider foaments, pharmaceuticals, whatever. Modify plants to be drought resistant, to produce vitamin A, really the precursor to vitamin A, and to make them robust for handling so that they don't bruise on the way to market, things of that sort. Modify animals to produce organs for transplanting. Pigs have about the same size organs as humans, so there's work underway to do that, and they've actually managed to do it for the external part of the cells of some of the organs so that they don't react. Some of the internals are not completely 100%, but there's movement in all these directions. And it comes with just a tremendous number of questions. Ownership rights, but they get patented. So you make this plant, and it's really neat, but it's patented. Contamination of other crops is one of the biggies. And in the early 90s, there was a herbicide-resistant soybean that was being planted, and a farmer that was growing soybeans in the next field found that his soybeans got contaminated with this gene from just pollen blowing over. So he figured, well, okay, I might as well plant them too. So he harvested his own soybeans and started planting them. And the company said, no, that's ours, you can't do that. He said, but you contaminated my fields. What do you mean I can't? So it went into the courts. And I think he lost, but they sort of paid him off a little bit. But those kinds of things get very contentious. And loss of biodiversity, long-term worldwide effects. And I don't know exactly whether I'll come up to it or not, but one of the things that has been coming out is that there have been sort of, well, let me finish with this one. Roundup Ready now, 1996 soybeans, corn, now also sorghum, canola, alfalfa, cotton, and wheat is soon. So all these things are going to be loaded with that. And if you're in a field next, whether you want it or not, you got it. And I remember they've already talked a lot about growing grass for grass seed down here, I think it's in Oregon, where they provide a lot of grass seed for golf courses, and they want a certain type of characteristic. So they're growing lots of it. And everybody's saying, wait a minute, that's in the open, and that's going to pollinate mine. I don't want your stuff. But there's really not too much that I've heard that people have been able to do much about it. Genetically engineered insect-resistant corn sounds great, and certainly some benefits. This is in Africa with specific place and situation in mind. So it works fine. Here they added beta-carotene to vitamin A precursor to rice, and initially they didn't have that much in the rice, but then they modified the rice so that they have more and more all the time. So now it's starting to be to the point where it's actually enough to give people a sufficient amount in their diet. One of the difficulties is that somebody owns the patent for this, and the trade-off I've seen recently is, well, if you're making less than $10,000 a year off of this crop, that's not considered commercial, you don't have to pay us. But if you're making more than that, then you've got to pay a royalty to us. Well, that's kind of iffy, and when are they going to change that? So it's a very sticky wicket. How? I'm sorry?
Do you have enough that we could invite you back for another talk?
Oh, I'm sorry. I couldn't see, it was in the dark. Okay. I think I'm just about done here. I wanted to talk about Andy for he's one of the guys they changed in genetic engineering. I guess I better stop off now. But I don't really have. You want a fluorescent cat? You can.
Come back.
Come back.
Okay.